So here is your digestive tube, okay, because your digestive tract is a tube all the way from your mouth to your anus. Okay, on the outside of the tube, furthest to the outside would be the visceral pleura, right? And then just to the inside will be smooth muscle, and inside of that would be more smooth muscle. Okay, so this is circular smooth muscle, and this is longitudinal smooth muscle. And just inside the circular smooth muscle is the submucosa, which is connective tissue, okay, primarily. And inside of that is the mucosa, okay? And the mucosa is made up of simple cuboidal epithelium if you're in your digestive tract. If you're in the esophagus, it's non-keratinized stratified squamous epithelium as it is in your mouth, right? So this is a tube of the, say, small intestine. Okay, so here's the lumen. Okay, this is your mucosa again. And just inside, within the mucosa, is this really thin band muscle called muscularis mucosa. It's really thin. The rest of this is connective tissue, which houses the blood supply. So it's kind of loose connective tissue in there, all it is. And in between these folds right here is called the lamina propria. And these folds are the macrovilli. Now, we learned this last semester, right? So this is not new. It's kind of a refresher review. Okay. So that's what we're looking at here. Okay, so here's your tube. And of course, in this particular tube, they don't have the folds like I was describing in my drawing. But certainly they have the mucosa, mucosa, the submucosa from here to here. And here's your muscularis mucosa. And then you have the plexus, the Meissner's plexus, or the submucosa plexus within the mucosa, okay? Or just on top of it. And then you have the first layer of smooth muscle right here. So this is your circular smooth muscle, and then longitudinal muscle on the outside, and then your serosa. Between the two smooth muscles, the longitudinal and the circular, is the Auerbach's plexus, planetary plexus. And you see these bulges in here? There's your sympathetic ganglion. Remember, sympathetic ganglion, or I'm not sympathetic, I'm saying that wrong. Somebody didn't catch me, how come? Parasympathetic ganglion, or in or on the organ of innervation. The sympathetic ganglion is a sympathetic chain going up and down the back of the thoracic cavity and abdominal. Remember that? And the superior and middle and superior, middle, and inferior cervical ganglion is part of the sympathetic chain. Ganglion and parasympathetics are in or on the organ of innervation. And that's what these are. There's your Meissner's plexus. Okay? So yeah, so there it is. And both these plexus participate in GI motility and secretion. Okay. So, I mean, sight of food and smell of food is the first step for the digestive process. So here are the salivators, the parotid, the submandibular, and the sublingual glands. These are the extrinsic salivary glands. In other words, intrinsic salivary glands, they're in your tongue and they're in the walls of your mouth. You have salivary glands all over inside you. Those are the intrinsic. We're not really going to talk much about them. Remember when we were looking at tonsils and there were salivary glands underneath them? Intrinsic salivary glands. The extrinsic are the ones that aren't built into our tissue, like our tongue. They are the parotid, the submandibular, and sublingual. So you have three of them. Saliva is 99.5% water, and the rest are solutes, like, what's in your blood plasma. sodium potassium chloride. There's some bicarb, some phosphates there, some urea, uric acid. Urea, uric acid in your saliva, that means you pee in your mouth, doesn't it? You can never say you've never tasted urea, because it's in your mouth every time you swallow it. It's an ultrafiltrated blood plasma. And there's urea floating around your blood plasma. So remember when you were a kid, you get a cut and you lick the blood? You're putting urea in your mouth at a higher concentration. In fact, blood really tastes like urea. It tastes like oil. I have, because my son peed in my mouth once. It really doesn't taste that bad. However, I wouldn't run around looking for something to drink. Unless I was in the middle of the desert. So it actually does taste like blood. In case you want to know. So, you know, you sweat urea too, right? We said this before. So every time you kiss your honey on your neck, you're sampling his urea. So the product secretes a very watery, and we said that already, and it has a lot of amylase. And amylase is the first step of digestion. The first step is digestion of starches. Starches are polymers of glucose. Okay? So amylase, salivary amylase starts breaking down starches so you can absorb it as glucose. The submandibular gland secretes food containing amylase plus mucus because it's a mixed gland. So is the sublingual gland. The submandibular and sublingual are both mixed glands. Okay? But the submandibular, and we'll talk about this, mixed meaning you're going to have mucus secretors and serous secretors. Mucus is mucousy and serous is watery. And we have serous demulum. So is what you have is this little cap on top of some mucus secretors. Mucus secretors stain light and serous secretors stain dark. Okay, so serous demulums is a cap of serous secretors on top of an acini of mucus secretors. So submandibular and sublingual are both mixed glands. The parotid is not a mixed gland, it's only the serous. So salivation is controlled by the autonomic nervous system via chemoreceptors and taste buds, and impulses. The inferior and superior salivatory nucleus in the brainstem, which in turn sends motor impulses via the facial nerve and glossopharyngeal, facial nerve and glossopharyngeal nerve to secrete. So the parotid gland histologically looks like this. It's all a dark color because, as I said a moment ago, serous secretors all stain dark. So this is staining. This is using a different stain, but it still holds true that the serous secretors stain dark and the mucus secretors stain light. So an acinus is this bulge right here. Okay, here's your acinus, and this would be a mucus secretor. This would be a serous demulum sitting on top of a mucus secretor, and they both secrete into this lumen. Okay, into this lumen, and eventually it becomes a duct, and you salivate out. Okay, into your oral cavity or wherever, out your oral cavity. So submandibular is a mixed gland, mostly serous. Sublingual gland is a mixed gland, mostly mucus. Okay. Okay. Here's your tongue, and your tongue is divided into the anterior 2/3 and the posterior 1/3. Remember, cranial nerve stuff. The anterior 2/3 is taste formed via the facial nerve. The posterior 1/3 is taste for the glossopharyngeal nerve. And you have taste buds in the back of your neck that's glossopharyngeal too, maybe some vagus. So you have these papillae on your tongue. And they're in different places. The valley papillae are the same as circumvallate papillae. Okay? So valley papillae has another name. You call it circumvallate. I like circumvallate better because it sounds cool. Okay? Circumvallate papillae. Okay. Anyway, the papillae that have taste buds are circumvallate papillae, folate papillae, fungiform papillae, but not filiform papillae, and taste buds associated with them. And the general design is like this. Okay, Here's your papillae, and you have creps in the papillae, and within the creps are taste buds. The taste buds have three cell types: gustatory cells, basal cells, which can give rise to either gustatory cells, or the third cell is a, well, not the third cell, the third cell is a sustenacular cell or supporting cell. So three cell types: gustatory, basal, and supporting. And basal can give rise to gustatory or supporting, so it's an undifferentiated cell. And remember way back when in AMP1, when we said all of the special senses, taste is a special sense, all of the special senses are mediated by epithelia cells. So these gustatory cells are epithelia. Okay? They're not neuronal cells. They synapse on a neuron. Okay? So there's your taste buds. Okay, the tooth, okay? The tooth is pretty straightforward. It has an enamel cap, which is 95% calcium. Okay? And then the cap is sitting on top of dentin, and dentin goes all the way through the tooth, right? And dentin is 70% calcium. I mean, dentin is more calcified than bone, okay? And the enamel is calcified more than both. So you have your gum line, and calcium, or the enamel goes and stops, goes below the gum line and stops just after it goes below the gum. And then the rest of the tooth is dentin. And the tooth is attached to the periodontal ligament, which is attached to compact bone. We call it alveolar bone too. And alveolar bone because of the alveolar margins, way back when, remember that? But it's really compact bone. So here's the different teeth. Who cares? I don't. We're moving on. Okay. So we're going to come back to this. This is where we're going to stop off. But before we stop, let's do a little bit of microanatomy. And then we'll call it a day. Okay, so here is a developing tooth. Okay? Don't ask me why my blind is good. Okay? So this whole thing right here is your tooth organ. Okay? And the pieces and parts of the tooth, well, check this out. This is an early tooth that hasn't even disrupted yet, and there's your gum line up there. So it hasn't even come out yet. Okay, so here is your stellate reticulum would be all of this area out here, okay? And you have ameloblasts that are sitting on top of what's going to be the enamel, and just below that are the odontoblasts, which are going to produce the dentin. There is a dentin line in here. You can kind of like see the dentin line going all the way around. Okay? Okay? I'm going to get rid of my writing because I don't want to save all that crap. Okay, so we're going to look at this area of the magnification in a moment, okay? Right there. Okay? So we have our stellate reticulum. So this is the top of the organ up here, and this is the edge of the organ on the bottom. Okay, same structures, just higher man. So here is your tooth. Okay. I'm sorry about this, but I don't want all this crap in my beautiful images that I spent so much time taking. Okay. So this shows, an artifact. Your gum is kind of separated from the tooth. That's artifact that's really stuck. And your gingiva, alias gum, is made up of non-keratinized stratified squamous epithelia. And there's your pulp cavity, and here's your dentin and your enamel out here. Okay, here's your periodontal ligament, okay, and you can see blood vessels in it. That's why when you bite down, it has some give. So here's your alveolar bone, and if you look closely, you can see that those are Herverian systems. In other words, contact. Okay. So here is, I didn't realize I didn't erase this stuff. Anyway, so here is your parotid, and I said earlier your parotid stays dark, and here it is, okay, and here it is at a high power. You don't see any foamy-like cells. Here is your seventh cranial nerve right there, okay? So your seventh cranial nerve goes right across your parotid. Your parotid is embedded in your cheek, and there's a little bit of that, folks. Here's a duct, okay? So then you look at another gland under low power. Most of this stuff in here is adipose. So this is the tissue we want to look at. You look at it a little higher power, you still can't recognize what kind of gland it is. Here's a couple ducts. Here's another small duct. Okay, higher power, then you can start to see the foamy-looking staining of the mucus-secreting glands, right? And here is a serous demilode sitting on top of the mucus secretors. And here's a little hole that they're secreting into. Okay? So this has to be the submandibular gland because it's mostly serous. Sublingual is mostly mucus. So this is most submandibular because it's mostly serous. And again, serous stains dark. Serous stains dark. Okay? And then you get in higher power and you can see the mucus light staining of cells. And these are 3 different slides, 3 different preps, 3 different lab tapes. So this is sublingual because it's mostly mucus. We get a higher power of it. And there it is. It looks better when the lights are turned off. I'm too lazy. Here is some more. What is this? If it's mostly mucus, it's sublingual. If it's mostly serous, it's submandibular. So I guess it's sublingual. Here's some more sublingual. Here's lower power, higher power, mostly mucus. So it has to be sublingual. Okay. More sublingual. Why? Because it's mostly mucus. Okay, here's some filiform papillae. And I said earlier, filiform papillae do not have taste buds. Circumvilli papillae, volar papillae, and pungiform papillae have taste buds. Okay? So this is, on this side, this is filiform papillae in an ungulate or a cat. Okay, I don't know what it is. A bunny rabbit in this case. Well, have you ever been licked by a cat? They have really rough tongues, filiform papillae. These filiform or cats, rabbits, and ungulates, they're actually keratin little hooks. Okay, we don't have them. Okay, our filiform papillae are not so fibry because we don't. Our epithelium doesn't get very keratin. Okay, so here are some pungiform papillae. Taste buds are right there. Okay, if you notice, this is a duct. So these glands down here are called von Ebner's glands that say salivate in between the papillae to continually wash away the contents so that you can experience a new taste each time. Put something different in your mouth. Here it is under higher power. You could say a pore, a taste bud pore. And here are some von Ebner's glands, and they're mixed glands. There's mucus secretors and serous secretors. Okay. And like I said, this is a duct going in between the 2 papillae. And these over here are skeletal muscle. I don't know why I do that. Okay. So here's different tissue prep. Here's some skeletal muscle. Here's some von Ebner's glands. Okay. Here is the remnants of the edge of a duct leading in between the papillae. And here's some taste buds. And it's non-keratinized stratified squamous epithelium on the top of the papillae as well. Here's more higher magnification of the muscle of the tongue, and you can tell it's skeletal muscle because of the striations. Striations created by the sarcomere. And it shows skeletal muscle going in every direction. That's why you can move your tongue down in every direction. Okay, right? You can fold your tongue. I can fold my tongue like this.
Yeah.
Uh-huh.
No, without the teeth.
Can you fold it the other way? Uh-huh.
Huh?
Wow. But some people can fold their tongue. Most people fold their tongue like that. And there's some that fold it the other way. I don't know how they do that. It's just the way the muscles of the tongue are designed. So here's the esophagus, okay? Non-keratinized stratified squamous epithelium. The upper third of your esophagus is skeletal muscle. And this is the upper third because on high magnification, you can see some striations. And the lower two-thirds of the esophagus is non-keratinized stratified squamous epithelia and smooth muscle rather than skeletal muscle, the upper one-third. Okay. So here's some more esophagus. This is a nice image. You have the mucosa, the submucosa, the intercircular layer, the outer longitudinal layer, just like everywhere else. Higher power, the same thing. Okay, here's your esophageal gastric junction. So here's your esophagus coming in, and then it changes to simple columnar epithelium in your stomach. Okay, so you got, this is higher power of this, and this is a different slide. This is just smooth muscle, what can I say? So later on, we'll start talking about the different cell types of the stomach.